And today we'll talk about the inactivation, removal, and regrowth potential of opportunistic pathogen and antimicrobial resistant genes in recycled waters. This research has been conducted with several people, including SA-Water, Paul Moniz, and David Cunliffe from SA Health, and John Turnage from A-Care, and of course, all my lab. So monitoring and managing waterborne disease is a critical aspect of biosecurity, and it plays an important role in minimizing the risk of infection disease entering, emerging, and establishing and spreading in Australia. This helps protect the Australian population, food security, and economy. Our biosecurity system is under growing pressure from the number of people and goods coming into Australia, and this number are forecast to further increase as soon as shown here in this slide in 2025. So globalization and the complexity of international supply chain and changes to the Australian operating environment, including those caused by coronavirus and the pandemic, are making biosecurity risk management more challenging. Nowadays, a range of technology are used to help manage this biosecurity risk. And during this presentation, we focus on the genetic tools used for water monitoring, recycled water monitoring, and I will briefly conclude with what we do with the monitoring of coronavirus in sewage. One important example is wastewater-based disease surveillance, and the World Health Organization use wastewater surveillance to uncover, for instance, polio outbreak and to assess immunization coverage in a resource-limited country. Because of the urgent need to monitor community widespread coronavirus, you will probably hear everywhere in the news, especially in Melbourne at the moment, wastewater monitoring has been extensively deployed to monitor the spread of infectious disease in a city, neighbor, important location, aged care, school, you name it, everywhere at the moment. So, wastewater-based epidemiology is extremely useful in responding to emerging public health catastrophe, like for instance, coronavirus, and antimicrobial resistance, or IMR. Antimicrobial resistance occurs when microorganisms change and adapt to resist stress at its impact, and the medication don't have any impact against them, so we cannot cure the disease caused by this microorganism. Without an action, it's estimated that in 2050, 10 million lives a year and a cumulative $100 trillion will be lost for antimicrobial resistance death. If you compare with what's happening at the moment with the coronavirus, so today we are slightly above 3,680,000 deaths, and IMR will produce 10 million deaths in 1 year. So is imperative that globally we enhance the antimicrobial stewardship in order to maintain efficacy of available antibiotics while seeking alternative therapy. This is very, very urgent, especially in the case of antibiotics that have been dubbed as last resort. All this label highlight this kind of antibiotic that are of vital importance. An example of how important is antimicrobial resistance at the governative level is the Australian National Antimicrobial Resistance Strategy document that was released early in 2020, which set the vision of Australia for fighting antimicrobial resistance in the next 20 years. So Australia, like many other countries in the world, are committed to protect the health of human, animal, and the environment through minimizing the development and spread of IMR. Is a very serious issue that is taken in extreme consideration also by the World Health Organization in order to avoid the future pandemics. So IMR is a quintessential One Health issue. The One Health issue is an approach which recognizes that the health of people are connected to the health of animal and environment. IMR has a clear link to each of these three domains, and as antibiotics are used, even if in a low level, in animal production, subtherapeutic dose of those are given as well to the aquaculture farms, and then we have farming, which are using fungicide and which create anti-fungicide resistance, and we have a wildlife who is actually getting in contact with these residuals, these pollutants created by the use of antibiotics, which are in the form of microbes. So microbes acquire genes that can express antimicrobial resistance and can subsequently be transmitted to humans. The pathway for the transmission are three, and the main three are from people to water and or the environment in general, and from the environment back to the humans, back to the animals, and back to the humans via food. So in this presentation, we will cover the first pathway, so the pathway between the people and the environment, and we will monitor the wastewater treatment plants to do that, because the wastewater treatment plants collect most of the residuals feces of human which have been using antimicrobials, whether in the community or in the hospital environment. And we will look at the transmission of antimicrobial resistance residuals from people to the environment, but as well as the environment may potentially contaminate the animals, and then from the animals to people. So one example of a critical antimicrobial resistance is the colistin resistance. And in here you can see a very famous manuscript led by Ling et al. In 2020, in which demonstrate that the use of colistin also known as polymyxin E in human medicine, has historically been restricted because of its toxicity. However, in recent years, colistin has become increasingly important as the last line of defense against fatal infection caused by multi-drug resistance organisms. So the identification of the first mobile colistin resistance gene, which is called MCR1 in 2015, triggered a rush of screening reports all over the world. Subsequently, we discovered 9 genes, so 9 kind of mutation of this colistin resistance, and that they were moving around the world via traveling. In this slide, you can see the first variant of colistin resistance, which is MCR1, that has spread mainly via people traveling in most of the world, and the other variant are slowly colonizing the world due to people movement. So antimicrobial resistance is quite a scary process that needs to be monitored. And the transmission is mainly via movement of people. To monitor emerging antimicrobial resistance gene, the best place to be or to monitor is the wastewater treatment plants. The wastewater treatment plants are also called the hotspot of antimicrobial resistance because wastewater treatment plants collect waste with, from farming animals, from industrial facility who produce antibiotics, from the population, from the hospital where antibiotics are used, and from all the sources available around an urbanized area and non-urbanized area. So basically what's happening in the wastewater treatment plant, all this sewage is collected and the microbes that are within this sewage are carrying antimicrobial resistant genes, and we can monitor the different portion of genes are emerging and which pathogen are still present or not, and quantify them. But also the wastewater treatment plant recently, they have been highlighted as being a hotspot for horizontal gene transfer, which is the mechanism through that allow the different microorganisms to transfer antimicrobial resistant genes between each other. So as I explained before, the hotspot means that we collect IMR, so we collect antimicrobial residuals in the wastewater treatment plants from all this environment, but also we use the wastewater treatment plants as a communication or as a pathway for the transmission of IMR to the environment. And I don't know if you can see my cursor, but this is the wastewater treatment plant. So once the wastewater is clean, is released into the environment or recycled. And part of this water carries antimicrobial resistant genes, which enter in contact with the water environment, for instance, and animals who are living in the water environments, they are in communication with other animals like wildlife, which is transferring the microbiome, or which is a fancy word for microbes who carries similar genes in this case, or a population of microbes. And they transfer to each other, and they transfer also to domestic animal. Domestic animal transfer to human, and human get back into the wastewater treatment plant, but also the aquatic environment and the farming of aquatic of fish and crustaceans, they can get in contact with antibiotics, and this can contaminate soil in which food grows, and the food, by growing, pick up antimicrobial resistance and then get in contact with humans via food. Or farming animal and treating them with antibiotics can also generate microbes who carry antimicrobial resistance. So it's a never-ending cycle, and there is different pathway. We won't cover all the pathway here, but the main pathway of communication between humans and the environment will be covered in this presentation. So how we sample the wastewater treatment plants. Here is us in action. And what we do the dirty job. So we literally go into the sewer and of course, we are fully vaccinated for this. In this case, we are the plumber vaccination. We have the plumber vaccination, and we go in there and sample either daily or either hourly. So sometimes we collect sample from early in the morning to late in the night to monitor population, and or we collect a daily composite sample to monitor other kind of characteristics of the water and wastewater treatment plants. So we also do different strategies. So we are also looking at the wastewater treatment plants as a place where antimicrobial resistance is potentially generated. And to do that, what we do, we don't only sample the influent, but we sample also the disinfection treatment of a wastewater treatment plants. And this disinfection treatment, they might generate or they might mutate a microorganism or facilitate horizontal gene transfer. So, and we are looking at each of them in order to find the place where potentially horizontal gene transfer is happening. We also sample the effluent, and both effluent, so the coastal effluent and the recycled water effluent. The recycled water effluent, for a purpose to look at potentially a pathway for transmission of antimicrobial resistance from irrigation water, and effluent is the pathway for us, the coastal effluent is the pathway for us to look for an IMR transmission into the environment. The main technique that we use to characterize, so to see who is there, what they are doing, and the diversity of the microbial community, we use this very complex workflow, which is the metagenomic workflow, and we use different pipelines and a different method of characterization according to what genes or what microbial community we are looking for. If we are looking for emerging antimicrobial resistant genes or plasmid transferred antimicrobial resistant genes, horizontal gene transfer, or simply the structure of the microbial community. So the first step for us was also to enter in contact to understand which are the emerging resistant genes, and which often they are not included in the database or in the metagenomics database available at the moment. So, and the emerging resistant genes are in Australia evaluated by the Antimicrobial Use and Resistance in Australia institute or entity, which discover the genes that are basically entering in Australia or emerging in Australia, or are not yet in Australia, but they are present abroad, and we have to monitor them in order to identify when a potential outbreak can happen or not. So we enter in contact with antimicrobial use and resistance from Aura, and we used, we share the same kind of assay for detecting the emerging resistance genes. And those assays are, will be here presented in this presentation. So first of all, let's look at the bacterial community structure in the wastewater treatment plants. So we sample different environments, as I mentioned before, the influent, the effluent, the sludge, and the intermediate treatments, which are not present over here. So as you can see in red is the influent population, so the bacterial community present in the influent. In blue is the bacterial community who is cleaned up from the influent and present in the effluent or coastal effluent in this case. And in pink, yellow, and green is the bacterial community present in the different sludge. So primary, secondary or activated sludge, and effluent sludge. So starting with the sludge community is very complex, as you can see. It contains several different opportunistic pathogens, and is not a major change. So there is a cluster of a community for each of the sludge, but there is no major changes or in the bacterial community. While in the wastewater treatment plant, a lot gets cleaned up, but there are part of the microbial community who survive. So this was a first step for us to understand, yes, we have microorganisms who are able to survive the wastewater treatment plants, and we have to dig in to understand what they are carrying and which genetic makeup they have. So we also perform shotgun sequencing, which is currently the most used method for us. And we try to focus on the opportunistic human pathogen and to see whether this opportunistic human pathogen survive the intermediate treatment of a wastewater treatment plants, and or they get cleaned up, they increase in quantity or not. And here in this graph, as you can see with the red arrow, are the microbial community who are increasing, so actually proliferating in the wastewater treatment plants. They are not very well-known opportunistic pathogen in this case, but in here, in the gray zone, they are the pathogen who are able to neither decrease, neither increase. So they are remaining stable. And these are Campylobacter, Staphylococcus, Listeria, and Salmonella, which may be opportunistic human pathogen. As you know, MRSA, Staphylococcus, methicillin resistance, Staphylococcus aureus is definitely an opportunistic pathogen, and Campylobacter jejuni is also a pathogen who is creating serious issue for people when they get infected. But the wastewater treatment plant in general is doing a very good job, so is cleaning most of the human opportunistic pathogen, as you can see here, like Klebsiella, Arcobacter, Escherichia enterococcus. So overall, the performance of wastewater treatment plants at the community level is very positive. So they clean the wastewater with the raw sewage, and there are some residuals of opportunistic pathogen, but most of them get cleaned up. At the gene level is a similar performance. So over here you have in blue the wastewater, then in pink the sludge, and in red the biosolid. So it's the secondary product of the sludge. And in South Australia, for instance, we use 100% of the biosolid produced from the wastewater treatment plants. However, this biosolid, of course, they get treated. So once they come out from the sludge, they remain, they are pile stocked to reduce the opportunistic human pathogen, or in general, the pathogen presence for years in the field. And then when they are considered safe, they get put in the agricultural field. So going back to the genes, so as you can see in the influent, there are a lot of genes present as expected, and then they get cleaned up. However, some of them survive, and by surviving, I will later quantify them, but they are surviving and they might be transmitted to the environment, for instance, in our coastal ecosystem. In the sludge, the profile of the antimicrobial resistant genes remain very similar, and not much is changing. But when we put the sludge in piled up for the production of biosolids, then definitely after 2 or 3 years that the biosolid are stockpiled, the genes are decreasing quite a bit. So overall is a good news. So the wastewater treatment plants are doing a very good job and in decreasing the load on antimicrobial resistant genes and pathogen. However, there are some residuals, and we try to focus on those residuals. And we focus on those. So now that we got the picture from the wastewater treatment plants, so at this level, we look at the pipe performance of whether the distribution system can interfere or can be a pathway for antimicrobial resistance of human opportunistic pathogen. And then we looked at as well areas when from this distribution system they might proliferate, for instance, a shower. And we also had a look at the irrigation of the reclaimed water dedicated to the irrigation or recreational use. So to do that, we performed again metagenomics analysis and shotgun sequencing, and here you can see a pipe, a distribution system, and we basically monitor different rooms in an aged care facility, and what we found out was in the different pipeline where the biofilm was present, and without extra disinfection, so without inserting an extra step of disinfection system, the load of bacterial load was quite elevated. And in here, there are the different colors correspond to different bacterial community. And then once we install an extra disinfection system, that in this case was an ECAS booster, we had a big decrease of bacterial load in each of the rooms. So we managed to actually clean electrochemically the microbial community. And in here, this is the plan of the aged care facility. So that's what was monitored. And they monitor also the air conditioning of the aged care facility. And what you can see, the Legionella level originally in the water that was involved in the air conditioning system was definitely high before inserting the ECAS disinfection system. And then after the installation of the ECAS, which is a booster for disinfection, the load of Legionella went down quite significantly. So there is a residual of pathogen. This is the message, that they are in the distribution system, and this pathogen, if they are not treated at the distribution system, they might proliferate. So this was a crucial point for us because we understood that the wastewater treatment plants are doing a good job. However, there are the residuals in the effluent and in the reclaimed water, and this residual might proliferate in the distribution system. So we had to find a way to understand the pathogen and also understand if they are proliferating or they might get in contact to human in a different way than the distribution system. So first of all, we separate our pathogens. So we understood that we had some common bacterial colonizing opportunistic pathogen, which we call it COP. So these pathogens are actually for us the most dangerous. And the reason why they are most dangerous is because they are, usually they don't create a great disease. Klebsiella pneumonia, of course, it does create disease, but they create disease in immunocompromised people or in a certain category of people. Sometimes they can live in us as a commensal. So commensal, it means organism that not necessarily develop a disease that you need to be hospitalized. Then we have pathogen that are simple opportunistic pathogen. So, and this pathogen, this reservoir of pathogen can be in, for instance, seafood, warm and coastal water, or contaminated water, fresh waters, for instance, Legionella pneumophila, or shower and cooling tower system. And then we have the frank pathogen, which are the pathogen that definitely cause disease, and they are transmitted via food, animal, and food products. So COPs are the common bacterial colonizing opportunistic pathogen, which are in our microbiome. So we have them all the time with us. They might and might not create a disease, but they are the one that we have to monitor more closely, because if they acquire antimicrobial resistance, they can definitely create outbreaks. And then we have the simple opportunistic pathogen, and this one, they live mainly in water and in raw or uncooked seafood or coastal water, and the frank pathogen, the food ones. The reason why I'm stressing a lot about that, because most of the people think that the human opportunistic pathogen are the one who are transmitting in the community, are basically creating disease, and they are the only one who are really dangerous. But the commensal one, or the colonizing opportunistic pathogen, are the one that can carry antimicrobial resistance, don't create a disease, but a certain be transmitted between human, and then at a certain point mutate and become multi-drug resistance organism. So are the one that we have to focus on, because this one create disease and they can be treated. This one are actually silent, and they might create outbreak or pandemic in the future. The simple opportunistic pathogen are easy to monitor, and they create immediately disease, and that's why they don't have a silent phase like the colonizing opportunistic pathogen, so they are much easier to control. So how did we monitor the pathway, and especially whether the COP were infecting us or were creating a disease, or were a problem? So what we did, we simulate basically a storage condition of reclaimed water for the common agricultural area, or we simply put the water in an oligotrophic environment, which means an environment which is poor in nutrient, and looked up whether the opportunistic pathogen were able to proliferate even in a condition which they don't have nutrients in the future. Why we did that? Because, as you know, the reclaimed water goes towards the irrigation water, and when it reaches the farms, it gets stored. Most of the time gets stored in open air, and when it gets stored in open air, what's happening? Birds and the environment gets in contact and provide nutrients. So technically, if there is a residuals in that environment, they can proliferate much faster. So what we did, we put the most strict condition which we have, and we wanted to see whether the opportunistic pathogens are growing and are carrying antimicrobial resistance. So definitely they are carrying antimicrobial resistance, and INTL1 is an indicator of horizontal gene transfer, and these are emerging genes, which technically they shouldn't be yet in Australia, and luckily they are not yet. And these are common variant of gene, beta-lactamase genes that they are monitored. And this is Klebsiella pneumonia carbapenemase, which can create severe hospitalized cases. And then we have the colistin that we talked previously, the 3 versions. So MC3, colistin MC3 shouldn't be in Australia, and it isn't yet. And MCR1, however, as I demonstrated before, is already in Australia, so, and is able to regrow and is able to proliferate after the wastewater treatment plant, so when the recycled water is cleaned up. In particular, KPC is able, the organisms who are carrying these genes are able to proliferate, so to come out of dormancy. This is a term that I'm throwing there, and I will explain later what it is. So the genes are cleaned up, but they are also proliferating. So they are able, once the recycled water goes out of the wastewater treatment plant, they are able to proliferate. How about the pathogens? So definitely the wastewater treatment plant concerning the COP pathogen and the opportunistic pathogen in general is cleaning them very well. Sludge is the place where they are most abundant. However, they are seasonally related. So for instance, the load of influent pathogen is quite high. And if we try to regrow in different seasons, and this is very relevant for from the farmer point of view, so we have a higher chance to have reclaimed water, contaminated reclaimed water in summer than in winter. And however, there are many important opportunistic pathogen or COP or commensal ones that they are able to reproliferate in the reclaimed water. Here is a picture of all of them, and as you can see, so it's a DNA and RNA based analysis I forgot to mention before, which means DNA is mirroring all the microbial community, so the dead and alive microbes, and RNA is mirroring only the active one. And what it means that some organisms are able to metabolically switch off, so go in dormancy once they go out of the wastewater treatment plants. And this is what it means. It means like the stress of the treatment of the wastewater treatment plants are they put them in a sort of emergency. So what microbes are able to basically build capsule around them and protect them, and they switch off their metabolism in order to be quiet or to survive the stress. And then once they are out of the stress, so once they arrive into the environment, even in oligotrophic condition, they are able to switch on, so get alive again. So from the dormancy condition that is creating in the wastewater treatment plants, they are able to resuscitate, is called, again in the environment, and proliferate. So we have a theory, and our theory is that our opportunistic pathogen in condition even prohibited for a microbe, they can be divided in 4 types. So the first type is a sensitive one, so it's a pathogen that gets into the influent and goes out and doesn't survive the disinfection treatment, and his quantity gets low and low, is very stressed by the wastewater treatment plants, and is unable to regrow. And this is a success from the wastewater treatment plant aspect, and also for us if it is an opportunistic pathogen. Some other pathogens are even more sensitive of that, so they completely die, or they get mostly removed at the 99% level. While we have some pathogens who survive through, for instance, a protozoa eating them, which is the phagocytosis, or in biofilm, or in dormancy. So they switch off their metabolism temporarily, or they protect themselves, so their quantity is not detected into the wastewater treatment plants, and then they repopulate and they go back to the effluents. And once they arrive in the effluent, they find a better condition and they grow again. There are others who are even more persistent, and they are able to completely dormancy, knock them down, so put their metabolism completely down, so they are undetectable at the wastewater treatment plant level and at the effluents, and then they get back again into the environment, and they keep on working and keep on proliferating. So this is a known phenomena that is very much related to the contamination of antibiotics into the microbial community. And it was never, or not much discovered into the wastewater treatment plant, but was previously discovered into the microbiome of humans. So what we are looking here is a normal phenomena, but that can be quite, that we have to keep an eye on it, especially at the reclaimed water level and in the food and waterborne pathways. Why is so important? Because, for instance, irrigation water goes to fresh food products, and by going to fresh food product, what it does, it basically sits into the leaf, but also plants are able to interiorize, so to bring the pathogen in, and some pathogen penetrate, whether via roots or via the leaf, via the stomata, and sit there. For instance, lettuce is one of those vegetables who is able to interiorize human opportunistic pathogen, which derive from irrigation water. And once we eat it, we can have a side effect if we eat a lot of lettuce, for instance. So how to trace a human opportunistic pathogen? I hope I still have time. It's via whole genome sequencing, and this we do it regularly in our laboratory. And what we do the One Health approach again. So once we discover the microbial community, once we identify a pathogen, what we do, we sequence the genome of this pathogen to understand whether it's truly carrying an antimicrobial resistant genes or not. And this process, we do it in collaboration with the Australian Centre for Genomic Epidemiology Microbiology, which is located in Sydney at the Technical University of Sydney. So we screen for several organisms, and these organisms are usually grown in several media, which are antimicrobial resistant or not. And the work is among us, of course, because we produce thousands and thousands of organisms in a few months. And what we do, we pick each of them. They look so pretty and beautiful. And we try to identify them and to sequence their genomes. One of the most common techniques that we use to identify antimicrobial resistance organisms is to grow them in selective media, and here you can see some of them. For instance, this one is a resistance to potentially resistance to carbapenemase. We are not sure until we sequence, or these are potentially resistant to cefotaxime. And so these organisms get to be sequenced, annotated, and sent around and identify their genomes. In here, we participated in a study which was covering all the influence, European influence, and we couple with some other countries in the world like US, Canada, India, and China, and South Africa. And we monitor all the influence for coliform, so for E. coli in particular, cefotaxime resistance. And what we found out that, first of all, Australia is doing very well because we have one of the best wastewater treatment plants at the effluent level, but we have also different, more disinfection treatments than a standard wastewater treatment plants. But we have less cefotaxime resistance coliform, which is a good news because it means we have less transmission of this organism. So to conclude, I don't know if Brad, I have time anymore.
We're running. How long is this last part? Sounds interesting.
Okay, yeah, to conclude, it's 2 minutes really.
Okay.
To conclude is what we do in our laboratory is the wastewater-based epidemiology of coronavirus. And so we participate in several studies doing that. And we are also in the Colossus node of Water Research Australia. And so we collaborate with all this university and water utility to monitor the coronavirus in the wastewater treatment plants. So, as you know, coronavirus is entering the sewer through hand wash, used tissues, sputum, and stool. So what we do, we simply try to detect, and in Melbourne is working very well at the moment, the area where there is community transmission. Prior, the community transmission is identified by in the hospital settings. So this is possible to our sewage monitoring, and to do that, we develop markers, not we particularly, but there were developed markers by the CDC who were able to identify coronavirus. And we can now detect also different variant of coronavirus in the sewage before they are detected by the hospital, in the hospital setting. In our lab, however, we use one technique because we mainly monitor in rural setting where the coronavirus is really low in quantity, if there is there, and we use the digital droplet qPCR. So what's the difference between the common qPCR, which is used for the wastewater treatment plant, and the digital droplet? That the digital droplet can detect a very, very low amount of coronavirus. So we can detect even one genome into the wastewater treatment plant. So it's very useful in a rural setting, not useful in areas which are heavily contaminated though. So to conclude, and I think I talked a lot, All of this is we do wastewater epidemiology is really a useful tool, especially is effective to monitor water and is essential for the Australian livelihood. But a safer water supply is critical to protect human health, the environment and the economy, including tourism and trade and agriculture. And we know that from the coronavirus pandemic. So it is crucial that we keep on developing system. This is a snapshot of the system that we use in our university, but there is more system that we can develop that support early detection, because if we don't detect early and we learn a lot during this pandemic and we are prepared for this outbreak, we cannot have an early warning and we lose a lot of money, we lose a lot of lives. And so we have to definitely develop better system, because the system that we have, they are okay, but they are not yet able to detect early enough outbreak. This will allow us to respond rapidly and efficiently to the future. And I know a lot of research is going in this direction at the moment, and the Australian government is investing a lot in biosecurity. So thank you. And this is it. This is our lab at the moment, and all the organizations who sponsor us and in our research.